Good evening, everyone. Welcome to the fourth session of Saliva Annex. Thank you for joining today. Well, let me introduce myself. I am Dr. Gargi Roy Goswami, and I am an interdisciplinary oral healthcare professional, a geneticist, and a general dentist by profession, and an academician by passion. And today, the journey of Saliva Annex, that is a free webinar series to propagate awareness about the magic fluid saliva through our academic endeavor of Indian Academy for Clinical and Dental Genetics, which is the educational wing of the startup named Croinas, the first of its kind in India, focused to deliver services and research promotion for saliva diagnostics and dental genetics. I welcome all of you. So before beginning the session, I will give I will give my gratitude and prayers to my dad, Dr. Kamalesh Chandra Roy, because whatever today I am and whatever I do, and the inception of Croinas is nothing but his inspiration. So he was the first dentist of a small town named Berhampur, district Murshidabad in West Bengal. That's my hometown district. And our dental clinic started in 1964 and it still runs very good. All right, not wasting much time. So let me go inside the session. As you all might have read about what we will cover today. So basically this session is on saliva and oral submucous fibrosis. We will try to understand in a very simplified manner the very complex oral submucous fibrosis. We'll also try to see a genetic perspective from how a normal cell goes towards pre-malignancy. Also, I will give you a simple tour into the molecular genetics of oral submucous fibrosis, and definitely I will give you some good insights on the different researches that has been happening throughout the globe, and few important salivary biomarkers which can we can think of in we can think more on that towards uh, developing any diagnostic tool for oral submucous fibrosis. And lastly, I will try to focus or give you some insights on whether we can use saliva as a possible diagnostic tool or not. So, what is oral submucous fibrosis? Um, we can abbreviate it as OSF or OSMF, right? Well, I'm sure many of you are aware of it. The very important and very nicely described and well-defined definition was given by Pinborg, and let me just read it out. An insidious chronic disease affecting any part of the oral cavity and sometimes the pharynx, although occasionally preceded by and or associated with vesicle formation. It is always associated with juxtaepithelial inflammatory reaction followed by fibroblastic change of the lamina propria with epithelial atrophy leading to stiffness of the oral mucosa and causing trismus and inability to eat. Oh my God. Did we understand anything? I mean, that's the way we generally used to mark up during our dental school days, right? We are not going to do that. Just I would like to tell you, just if you see my slide and I have highlighted this portion with yellow, juxtaepithelial inflammatory reaction followed by fibroelastic change of the lamina propria with epithelial atrophy. We will come back and see what it is. Well, before that, can you recognize this person? Any idea? No? All right. That's Professor Pinborg. the definition we just read about, I mean, it was given by him. So let me go to the next slide. Fine. So as you see that this is a very typical representation, a very simplified representation of oral mucosa. Oral mucosa as in the covering of our cheek, the inside portion that faces towards our teeth. All right. So what we have is here these pink colors, as you all understand, that it is oral epithelium. This is epithelial layer, and as you see that there are so many things. These globules are kind of adipose, and as you see, this is lamina propria. Just now you read in the definition, and these are to do with some fibers, some different types of cells, and this is muscle, and this is submucosa. So, can we revisit the definition once more? Yes. So, it's juxtaepithelial. Let me just simplify. Juxtaepithelial means is near to the epithelium. So, that means juxtaepithelial inflammatory reaction. So, some problem is going on which leads to inflammatory responses in the somewhere near the epithelium, and that's the stroma, nothing else, in the lamina propria, and followed by fibroelastic change of the lamina propria. So something to do with the fibers that are present in the lamina propria, and with epithelial atrophy. Atrophy as in degeneration. So what we understand is that oral submucous fibrosis, or OSF, is nothing but some inflammatory reaction that is happening, which causes uh, some kind of fibers to problem with the fibers that are present in the lamina propria or connective tissue, however you tell it, or the stroma. And because of that, there is also atrophy or degeneration of the epithelial layer, epithelium, like as in the cells becomes dead and they start becoming keratinized. So that is what is the definition of OSF says. I hope it got little simpler, right? Fine. Now, a very interesting thing I will tell you now. So next, um, is that to understand um, why a normal cell becomes premalignant. How is it? See, for example, premalignant or precancerous, as you say. If you see this figure, this is absolutely the cursor where it's moving, my cursor which is moving, it is absolutely a normal cell, epithelium rather. Now, in this initiated one, there is one kind of red dot. So that is a cell which is going wrong. Now, precancerous or premalignancy is a condition which doesn't happen in one day. Cancer doesn't happen in one day, right? Uh, so it happens with some time. It takes some time to go ahead from normal to abnormal. So precancerous is something, or premalignant cell, very simply we can tell, is something, a kind of abnormality happening in the cell, or it has abnormality that has initiated inside the cell. All right. So a very important factor that a normal cell becomes abnormal or premalignant or precancerous, however you put it. So there must be some agent. So you know that we all know that there are many cancer causing substances, and particularly when we talk about OSF, or oral submucous fibrosis, betel nut and areca nut. Yes, betel nut or areca nut is the one, and also there are other many reasons about it, but behind it. So whatever it is, so there is a latency period. So that's the period of time uh, between exposure to a carcinogen and later development of cancer, which is very important to uh, when we tell about normal to pre-malignancy. Like for example, you might have seen that many people tell that, oh, I have quit smoking almost 20 years back. but they suddenly develop cancer. And they are surprised that I developed a lung cancer, but I have left smoking 20 years back. The problem is that 20 years was the latency period because the carcinogen present in the smoke, whatever it is in the cigarette smoke, the oral mucosa got exposed slowly. It's like a slow poisoning for those 20 years, and then slowly the mutations in the DNA of those cells, some problem happened, and that's why over the time the cell became precancerous or cancerous. One important thing with pre-malignancy is that when a normal cell is initiated to become pre-malignant, or say it becomes pre-malignant, doesn't mean that it will always be, it will always land up to cancer or malignancy. No, because the particular initiated cell, I term it like that, the initiated cell, that means the cell which has started getting abnormality, um, also can be exposed to certain good environment which helps that cell to revert back to normal. So, few important points about pre-malignant epithelial lesions were shared by Dr. Michael Sporn, who is known as the father of chemo prevention event. And these include as being diffuse and multifocal, having a statistical or probabilistic nature in progression to malignancy. That means there is a probability that a pre-malignant cell would become cancerous. And mind the number 3, enhanced, that is increased DNA synthesis, and as well as mechanisms of protection. Whoa, I think we are much familiar with this picture. This is something called as histomorphologic changes of epithelial dysplasia. Okay, what's that? Dysplasia is any change in the cell is dysplasia. It can be the nucleus size that changes. It can be the shape of the cell that changes. So when we tell histomorphologic, that means histo relates to tissue. So we are talking about epithelial tissue. Morphologic, the way we see it under the microscope. So it denotes size, shape, etc. So I think if we see this picture, which is a very classical type, and we see that from normal it goes to initiated, then so on. So there is one culprit cell which then provokes the others to become abnormal, and slowly this increases, and finally we see that something goes wrong, and the in epithelium itself seems to come down, and we have definite features which we study a lot, like drop-shaped ridges or nuclear hyperchromacity. That's because we have enhanced or increased DNA synthesis, increased mitotic activity, yes, because the cell starts abnormal growth, abnormal multiplication, and proliferation. The problem is if all these whatever points that you see, the histomorphologic changes would have happened in a single lesion, it would have been very easy to identify. But rarely does one lesion have all of them. So let me tell you one thing. Okay, see this picture. Just observe. Do you see anything different in this image? Yes, right? Something, not much different, but something different in the central part. So that means that the central part of the image, if I consider each of the squares with the other, which are outside the central part, the squares seems to be not very different from each other, but as a whole, if I see the central part, yes, definitely this subset of boxes has gone something weird, or it's doing something different compared to the other squares around it. And it also seems that this particular subset of central boxes are in some relationship with standing relationship with the other boxes. So what I'm trying to tell you is about a very important theory called tissue organization field theory. So this key factor in this theory is that the cells are not independent units. It is always a part of a larger assembly. So to understand a particular disease or the genetics or the molecular expression, we cannot consider if we just study one individual cell with individual gene expression. It has to be understood in the form of a network. So tissue organization field theory says that the progression of normal healthy tissue to a precancerous and eventually malignant state is primarily a problem of tissue organization. Herein comes the concept of microenvironment. As I was saying, that is just if you see the boxes which are around the central part. So basically, this microenvironment or the cells, different types of cells that are surrounding a particular in a tissue, a particular area. So if they're getting exposed, by any carcinogenic agent, so that might destroy the cell-to-cell recognition. That might compromise the genomic integrity, and these gives the base for DNA mutation to happen, for genetic mutation to happen more and more. That is accumulation of mutations. So. As in all, we know that most cancers happen in epithelial tissues because those are the cells that divide most frequently. And as you see that epithelial cell will never grow alone. It needs a substrate. It will need a connective tissue, an extracellular matrix, and other cell types called stroma. We can very well see that. So according to the theory, the cancers can arise in epithelia by way of disruption because there is continuous communication both ways with the stroma, extracellular matrix, with the cells, with the epithelial cells. So, if a carcinogen could distort the interactions between stroma and epithelium at the level of stroma, and epithelium then goes nuts and proliferates to produce a precancerous mass. This the picture we see here. One cell, more cells, and more culprit cells, and then it goes really, really bad. So this disruption of tissue that actually starts the abnormal growth is actually preconditioning or is actually the platform for accumulation of mutations. That is what I wanted to emphasize. So to understand why this DNA mutation happens, or rather how it is that the mutation is happening. So is it that if mutation occur, then everything becomes cancerous? No, not at all. For example, mutations in DNA can happen for many reasons. For the chemicals that are contained in areca nuts, we know, right? So we're talking about OSF, so I give the example of cigarette smoke or areca nut. So these chemicals will cause DNA damage. And in most cases, the DNA damage will not lead to cancer or the disease. But in some cases, it might. See, for example, each of our cells has more than 25,000 to 30,000 genes. And we do get mutations, but that doesn't mean that it develops to a cancer. But if the mutations occur in some key genes, then definitely cancer develops, or rather a normal cell becomes pre-malignant, and then if more mutations are happening, it goes to malignancy and bad cancers, right? So these key genes can be classified into basically three groups. First one, growth promoting genes, also called as proto-oncogenes. So what these do? They normally tell the cell when to grow and divide, and it's just kind of instruction they give. So you have to grow now, you have to divide now, stop, please stop. So they give the instruction to grow and divide. These are normal genes which are present in us in the cells. Then the second group is growth inhibiting genes, also called as tumor suppressor genes. Their normal function is to maintain the cell in a non-dividing state. So if proto-oncogenes cells a cell to grow and divide, tumor suppressor will, after some time when the need is not there, tumor suppressor will help the cell to stay in a non-dividing state. Another group of genes is DNA repair genes. So these genes function to repair any damage happening to the DNA. That is why it's an inherent mechanism that we have inside our body, which in spite of that we get mutations. Again, it reverts back to normal because of DNA repair mechanisms or DNA repair genes. Now let us take one by one. So proto-oncogenes becomes oncogene if there is a mutation. So what are these oncogenes? So oncogenes will now, basically it's a mutant form of proto-oncogene, and it will actually induce cancer. And in most cases it is seen that the members, I mean the oncogenes are members of signaling pathways, all right, and mostly they are growth factors because proto-oncogenes, and it is just a mutant. So what is the normal function of proto-oncogenes? It's involved in growth or the control of growth. So if it is mutated and it becomes oncogene, then definitely there is a deregulated cell division. So in the process of converting proto-oncogenes to oncogenes, let us see what can happen. These genetic changes that happens and converts proto-oncogenes to oncogenes will lead to either increase in a protein product, or it will change, there will be change in the activity of a particular protein. It suddenly can become a vital protein can become non-vital, or a very important protein can just go off because it is not forming. So to basically tell you the important mechanisms, okay, the main mechanisms, genetic change mechanisms that converts proto-oncogenes to oncogenes, that is how a cell goes from normal to pre-malignancy: chromosomal translocation, gene amplification, point mutation, and some local DNA rearrangements. I'll try to simplify it with this. So this is a proto-oncogene. What is translocation? So one part of gene from other part of the chromosome comes and sits on the head of another gene, and that then it changes. All right. If you see promoter is a part of DNA that controls the genes. All right. So it will then stimulate more growth proteins. Gene amplification. In this, what happens in this mechanism, a gene will be copied number of times. So if the gene was producing one protein or one growth stimulating protein, now it will, if it is three copies, then it will produce three. So there is an excess again. Now another is point mutation. If a point mutation occurs in a promoter region or the control element, then again there will be some problem with the particular gene, and it might lead to again the normal growth stimulating protein in excess. And what I mean by local DNA rearrangements is that the point mutation can happen within the gene itself. In that results either in a hyperactive protein or a protein which cannot be degraded, which might be earlier was degraded in normal thing, but now it is not degraded. So these are the different genetic mechanisms to be very simple to represent in a very simple way, which converts proto-oncogenes to oncogenes, and thus a cell moves from normalcy to pre-malignancy, and then further to malignancy. Second group is tumor suppressor genes. So in cancer, the cancer can arise if there is a loss of tumor suppressor genes. It happens so that their normal function was to restrain cell proliferation. Now, if there is loss of tumor suppressor genes, that means the cells will not stop dividing or growing. So if you see the list there, these are certain common tumor suppressor genes that are present in different locations in our cell with different functions. DNA repair genes, as we know that they will function to repair DNA damage. So any defective DNA repair can contribute to cancer because this again allows more multiple mutations to accumulate to cause the disease state. So, for example, if there is we have, in OS particularly, there is reacting. We know that this aricolin causes reactive oxygen species. Then that is oxidative stress. So that might cause DNA damage. Now, in case if the repair mechanism is fine, then it will be repaired, or it will be growth arrest or senescence and death of the cell. But if it goes on, there will be mutations and rearrangements and more active oncogenes, inactive tumor suppressor that leads to the malignant form. Well, that was a genetic perspective of normalcy to premalignancy. Now coming to the molecular genetics of OSF. I have put a very simple flow of representing the pathogenesis of the oral submucous fibrosis, and we know that it is a multifactorial disease. So let us take out the most important feature cause, that is areca nut, and we know that these two things would be related to the duration frequency of the habit, that is the constant irritation and the exposure of the oral mucosa to these. Also, if there is deficiency of iron or vitamin B12, it also increases susceptibility to the particular OSF, and there will be chronic inflammation, and we have already understood from the definition itself. So there will be recruitment of immune cells, and there will be increase in growth factors like TGFbeta and increase in cytokines. All right, let's move on to the next. I have taken areca nut, and we know that areca nut is composed of alkaloids, flavonoids, trace elements. So when we talk about molecular pathogenesis of OSMF, it basically involves collagen disruption in the collagen metabolism. Because if you see the alkaloids, for example, the substances in areca nut, that is alkaloids, and among them there are many alkaloids which have been identified. I just take ericolin. So what it does is it basically induces collagen formation. And if I take polyphenols, for example, in that the flavonoids will just inhibit collagenase. So collagenase is an enzyme which breaks collagen. So that means it is suppressing collagen degradation. That's why more collagen would be formed. And also another trace element, very important, of quite important, notice is to of about copper. So copper also upregulates an enzyme called as lysyl oxidase. So this enzyme is basically required for um, cross-linking of collagen, because collagen is a big molecule. So we'll see how it is happening. So to give a simplified view, what we do is we understand the genes that are being activated uh, in the collagen production pathway and those in the collagen degradation pathway. So in collagen production pathway, there will be activation of pro-collagen genes by TGF-beta. So I take TGF-beta, this growth factor, because it has two-way role to play. We'll see one by one. Then there will be elevation of pro-collagen proteinase genes. That's why these enzymes, pro-collagen C proteinase, pro-collagen N proteinase, would be elevated, and there will be upregulation of lysyl oxidase gene activity. So more of this enzyme. And in the collagen degradation pathway, there will be activation of tissue inhibitor of matrix metalloproteinase gene, that is TIMS, and activation of plasminogen activator inhibitor gene, PAI. So we will just remember the short forms. So because there is a massive involvement of genes and growth factor interactions, which involves collagen production pathway as well as degradation. So OSMF can also be regarded as a collagen metabolic disorder. Before going into more details, this is a schematic representation of formation of collagen. Okay, don't get confused. I mean, collagen is a complex molecule. So this is just that it starts in the rough endoplasmic reticulum. So pro-collagen chain synthesis and modifications happen, and then there are formation of bonds. There is alpha 1, alpha 2 chain, blah blah, and then basically collagen is a triple helix, three different chains joined together. So that's triple helix, and this is the form called procollagen. It's like kind of primary collagen, not the final one. So from this, then it moves to the extracellular environment, and there you have all those proteinases which we just talked about, and they help in cleavages, enzymatic cleavages at the N-terminal and C-terminal. Well, if you remember your biochemistry, so N-terminal in any protein is made up of amino acids, and each amino acid has amino group and carboxylic group. So in that way, the enzymes that cleave from the amino side, so it's N-terminal side. So that is how it is N proteinase, and from the carboxylic or C side is C proteinase. All right. Then we have lysyl oxidase, that is LOX, and then the collagen. Between the collagen fibers, there is cross-linking, and you get that tough and stiff collagen. So let us see that if I take exemplary canut and I say that I'll be taking TGF-beta, so what it does, it upregulates certain procollagen genes. It upregulates the TIMP1 gene expression, also lysyl oxidase, and it downregulates MMP1 gene expression and autophagy, and it again upregulates epithelial mesenchymal transition. I will talk about it, which is a very important thing. All this leads to submucosal fibrosis, and that is oral OSF. So TGF-beta, we first take the collagen production pathway. So it activates procollagen genes like COL1A2, COL3A1, COL6A1, and so on and so forth. So resulting in production of more procollagen, right? Increased more procollagen. And it also activates procollagen C proteinase and procollagen N proteinase enzymes. And all this leads to, we know the function of these enzymes now, and it leads to increased collagen formation. Also, on the other hand, if you see that the PCP, that is the procollagen C proteinase, also helps the conversion of prolox to LOX, that is lysyl oxidase. So this again here from ericanut comes in copper, which has a good role, and then all this together leads to increased collagen formation, increased collagen production. Now, the other thing that we were seeing is how TGF-beta activates the gene for TIMP, that is tissue inhibitor of matrix metalloproteinase gene. So tissue inhibitor of matrix metalloproteinase gene. So that means matrix metalloproteinase is nothing but, proteinase means it will break down, right? So break down some proteins. Proteins of what? Matrix. I'm making it that way. So let us understand it in a very easy way. So that means there is some enzyme which can degrade the collagen matrix, that is collagenase. So if I have to go to the disease, get the disease, OSF, then I need more collagen. That's the only goal. So I have to inhibit collagenase. That is what happens when TIMP gene is activated, because more TIMPs, more collagenase is deactivated. Okay. Similarly, activation of PAI, plasminogen activator gene. So the plasminogen activator inhibitor protein actually converts plasminogen to plasmin, because plasmin is again required for conversion of procollagenase to collagenase. But if plasmin is not formed, then again collagenase will not be formed. So that leads to decrease in collagenase activity, and this is also supplemented by flavonoids in the ericanut. It also inhibits the collagenase activity, and thus there is a decrease in collagen degradation. Ultimate result, more of collagen formation, that tough, stiff muscles, and resilient form of OSF, and that leads to all the symptoms which we know classically present with OSF. If I don't talk about epithelial to mesenchyme transition, doesn't end. That wouldn't be a justice. So what is this epithelial to mesenchyme transition? That's a process. If you remember the first slide where we showed the different types of dysplasia, the last one was showing that it starts migrating in, going down inside breaks the epithelial barrier and goes down into the stroma or the lamina propria, whatever you say. Why that happens? Because there is a change, massive change. Epithelial cells, as if they change their shape to mesenchymal state. So this is called epithelial to mesenchyme transition, where the epithelial cells converted to migratory mesenchymal cells. And the most common thing is the loss of E-cadherin. All right, so E-cadherin, to be very simple, if you see this in the picture, there's something green. These are basolateral markers, basically. So basolateral in a sense that they keep the cells in shape by binding with the cells to each other and also with the basement membrane. So if this green is lost, then the cell shape changes. Just like in the figure, just it becomes spindle shaped. So now they are able to move down, and in the process, in this epithelial to mesenchyme transition, there are many repressors that are activated, because E-cadherin being not formed means it is promotion of EMT, that is epithelial to mesenchyme transition. So for example, you've seen some names here: snail, slug, zinc finger, E-box binding, ZEB1, twist. So these are E-cadherin transcriptional repressors. Very easy. E-cadherin is a gene, all right? So this green color is the protein, right? So for getting this protein, the gene has to be activated. Now in OSF, what is happening? There is this epithelial to mesenchyme transition is happening. So for this, the cell don't want E-cadherin, so it will repress. So these transcriptional repressors means that these are different moieties, which molecules, to be making it very simple, which goes and binds that particular gene and doesn't allow the RNA polymerase to transcribe, stops the process at the gene level itself. That's why it is called as transcriptional repressors. There are many intricate details inside, but I'm not going into that, and that doesn't make any sense. All right, so that is the most important aspect of, to give you in a nutshell, about molecular genetics or molecular pathogenesis of OSF. Well, this is a very good list which says about different types of OSF biomarkers, like trace. It has divided into trace elements like copper, iron, according to the different researches that has happened. Antioxidants like superoxide dismutase, vitamin C, vitamin E, immunoglobulins, oncosuppressor genes are the genes, etc. Now let me just tell you about certain oral OSF salivary biomarkers. See, there has been ample work. It has happened, it is also happening. Thing is, there are many markers which has come up now, but it is still yet to be deciphered what is that pinpointed biomarker. And I feel there will not be one single one, because we have to think of forming a panel, maybe, because OSF itself is a very multifactorial disease, a polygenic disease. So it's not the work of one gene going wrong. Well, so the most important ones is, like in OSF patients, it has been seen that there is elevated DNA damage, and whenever there is DNA damage, and there is lipid peroxidation levels are high, because that means there is DNA damage can occur only if your cell membrane is getting disrupted. So cell membrane getting disrupted is definitely, you are just opening up the lipid bilayer. So that's how your lipid peroxidation levels are high. So one such lipid peroxidation marker, very important, is malondialdehyde, MDA, and it is a very important marker with which many researchers have shown that it says about whether a OSF patient has, it says about the malignant potential of that particular OSF condition. And also another one is 8-hydroxy-2-guanosine. So this is also again due to DNA damage. When DNA damage, these are all DNA adducts, different chemical modifications occur because of the intervention of these carcinogenic compounds, and the structure changes, the nucleotide changes, and they get this weird structure and weird name too. So you can detect them in saliva. So salivary 8-hydroxy-2-guanosine is again a very important marker. And there are other markers like vitamin C and vitamin E, which were found in different studies to be lower in OSF patients and compared to healthy controls. And to mention few more, salivary dismutase, salivary superoxide dismutase and glutathione peroxidase. These are also certain researchers, certain papers says about it. And basically they are downregulated in saliva. And salivary S100A7, this is a small calcium binding protein, and it has been associated with development of carcinomas in different types of epithelial as well as development of psoriasis. So high expression of this particular molecule is observed in potentially malignant disorder. In that way, we can connect with OSF and understand whether a particular condition has high malignant potential or not. Okay, coming to the last part, can saliva be a diagnostic tool? So what would be our obvious answer, or what would be our answer rather? So we all will be telling yes, because saliva is very versatile, because its collection is undemanding, the procedure is non-invasive, there is no trained medical staff needed, multiple samples can be obtained, it is safer to handle, minimal risk of cross-contamination, and so on and so forth. But we have to understand that saliva is a very complex mixture of different types of substances. It also has unique biomarkers which we need to find out and panelize for OSF in particular. Also, we have to, while selecting the biomarkers and to use them as a diagnostic medium, we need to understand what we are selecting. Are we going to select DNA, antibodies, proteins, lipids, metabolites, etc.? So that will change the directions of the study. Another very, very important aspect that has to be understood whenever we are trying to analyze a biomarker or we are thinking about a salivary biomarker, say related to OSF only, it is applicable to any disease in that matter. We need to understand how this molecule is coming from blood to saliva, because ultimately saliva is just a kind of an extract out of blood of the plasma. It's like a watery secretion more. So that's why whatever is there in the blood or plasma is there in the saliva. That's true. But again, it depends on the mechanism. The amount will differ. So for example, mostly it follows diffusion, active transport, filtration, and etc. So before even we select a biomarker, we need to understand how that particular molecule actually has come to saliva, which mechanism it has followed. Now, being overwhelmed with the versatility of saliva is not a very proper thing. We need to understand the unique challenges with saliva before we think about telling that yes, saliva can be a diagnostic tool for OSF or any other disease that matter. So first of all, concentrations of specific biomarkers much lower than compared to plasma. Confound specific oral cavity, that means, for example, it's mixed with food particulates or drink or residues. The changes in salivary flow rate about stimulated and unstimulated saliva. Also, use of preservatives. Sometimes they're used for stabilization of certain biomarkers, but that can again invalidate certain methods of your analysis. A very important confounding factor is viscosity. Very important. Now, viscosity can also include the presence of bubbles. That is another where while collecting the specimen, viscosity will vary from participant to participant, individual to individual. Also, viscosity will again vary depending on the state, whether in exercise or stress or hydration status of the participant. So, and for finally, it is also important to understand the challenges of selecting collection method and understanding the available collection devices, whether to use them or not to use them, where to use them, when to use them, how to use them. Because stimulated and unstimulated saliva, for there would be significant differences in concentration of the analytes that we want to quantify. So it is very important to determine both the most viable collection method and technology required. And if I am thinking to develop something for myself, I also have to understand what I want to do with that test. Do I want to do a disease, or I want all of this? Second, whether I want to develop a diagnostic tool which would be qualitative in nature or both qualitative and quantitative in nature. That means the design of the work plan or defining the work plan is very, very important. Right from classifying the study groups and control, selecting the biomarkers, it happens mostly in combinations, and methods of analysis, whether you are going for simple spectrophotometric analysis, whether you should go for ELISA or more high-end like mass spectroscopy, etc., etc. Well, with that, this is my husband. He's my backbone. I thank him, and I thank my mom for bringing me into this lovely world. And these are my wings, Shruti and Kanchan, and others who helped me a lot in my passion, in living my passion. They live my passion. And this is my lovely family and my beautiful friends who are always with me in whatever I do, and my bro buddy, Dr. Anirban. And I celebrate life very much, and you please do too that. And please follow us at Instagram, #Croyness, in LinkedIn, as well as in Facebook. To have more fun with science and life, please join our Facebook group named as Indian Academy for Clinical and Dental Genetics. We do a lot of stuff there, and we do live sessions on Wednesdays, discussions, and also we have something called as Showcase Sundays, where you can come and promote your talents, your products, your courses, or anything that you want to speak to the entire world. Thank you so much for joining me today, and I hope I will meet you again very soon. So with that, I end today's session, and I hope you have enjoyed. So if you have any questions, I can take it up. And if you have liked this session, please give me some thumbs up. It will inspire me. And, my next sessions will be, I hope, I'll try to make it more interesting. And please subscribe our YouTube channel. So with that, I will end, and be safe, stay safe, and take care, and be positive. We will overcome this difficult phase, and don't worry, be in health. That's most important. Thank you.